Good evening. Uh, Good evening, Maha. Are you able to hear me? And is the screen visible?
Yes, ma'am.
Okay, thank you. Okay, so as of now, only you have joined and welcome you to the session, like week 7 live practice session for environmental biotechnology course. This week, we will be again dealing with the methods. We'll be continuing from the previous discussion. I hope you are comfortable with the question and answer session. So I don't think you will take much time in answering. You can just think and answer. Today also we'll continue with the same question and answer session. Is it fine with you? Okay. And if you have any specific doubts, you can also post that uh, now or later. You can just post. So I have some slides on this uh, methods. Do you want to go with the question first, or do you want to first have a little overview and then go with the questions?
Questions, ma'am.
Okay, then you can start answering. So for assertion and reasoning questions, you can just give if both the statements are right and if the reason is explanatory, or if any of it is wrong, or if both are right and it is not explaining. So from these options, you can answer all the assertion and reasoning questions. So DGGE stands for denaturing gradient gel electrophoresis. Okay, so the answer is right. DGG stands for denaturing gradient gel electrophoresis, and in this we will be having a GC clamp at the end of the DNA fragment. So GC is guanine cytosine, and in a DNA always A pairs with T, adenine with thymine, and guanine with cytosine. So but the AT pair is bonded using a double bond, and the GC pair is bonded using a triple bond. So that's why breaking of GC bond is a little difficult than breaking the AT bond. So that is why we are using a GC clamp in DGGE, and in as the name suggests, we will be running a gel electrophoresis. So electrophoresis technique we use for separating molecules based on their size. So in this, okay, so I hope uh, the connection issue is a bit resolved now, is it? Either of us has have joined again. Like, is my voice clear now? Or okay, I'll anyways give the explanation and then move to the next question. So whenever you find the next question, you can answer for it in the chat box. So as I was telling, the GC bond is a triple bond, and hence breaking the GC bond is a bit difficult. So we will be having the DNA fragment clamped using a GC so that we don't completely denature the double stranded DNA, and then it is a gradient electrophoresis. So we will have different concentrations of denaturing agents which will break the DNA as the electrophoresis moves on. So based on the sequence, the denaturing will happen, and because of that we will get the DNA bands at different locations in the electrophoresis. So this is the principle of TGG, and your answer is right. So the next question is whether the functional marker genes can provide information about community metabolic potential. The reason is only ribosomal genes are suitable for environmental community analysis. Okay. So functional marker genes can provide information about community metabolic potential. That is true. Uh, because functional marker genes means these genes are expressed and are functional. Like from the DNA we are getting RNA transcript which is then translated into protein. So we that genes which are translated finally into protein we call as functional marker genes. uh, we can usually find it using the RNA analysis, and they provide information about the community metabolic potential. What is the metabolic potential of the entire community present? What kind of substrates they can utilize and what kind of products they'll be able to produce? That is the metabolic potential. So functional marker genes can provide information about community metabolic potential is true. Uh, But the second statement is only ribosomal RNA genes are suitable for environmental community analysis. This statement is false. Like apart from ribosomal genes, like mentioned in the first statement, functional genes can also be used for environmental community analysis. So both functional genes as well as ribosomal genes can be used. It's not that only ribosomal genes are used for environmental community analysis. So here the assertion is true and the reason is false. And the next question is PCR, which is the polymerase chain reaction, is essential for community DNA fingerprinting methods. The reason that is stated here is PCR amplifies the target genes, allowing analysis of otherwise undetectable population. So here, community DNA fingerprinting, it is a molecular technique. We generally use it for profiling the microbial community diversity. Uh, at the end, we have a simplified genetic fingerprint from the DNA analysis. So we perform the DNA analysis and uh, we are just uh, profiling what kind of diverse microorganisms are present in the community. So this is community DNA fingerprinting. And PCR is it is stated that PCR polymerase chain reaction is essential for community DNA fingerprinting. And the reason is because PCR amplifies the target genes, allowing the analysis of otherwise undetectable population. So here comes the concept of culture dependent and culture independent molecular techniques. So we have discussed about the culture dependent techniques where we have to run laboratory experiments to culture the microorganisms, but as we have seen in the last class, very less amount or uh, very low percentage of microbial populations that are present in the nature can be detected using culture dependent method. So that is why we were in need of culture independent method, and we have shifted to many other analysis like genomic analysis. In all of these, one common step is the polymerase chain reaction. So the polymerase chain reaction is generally used to amplify the genes to get multiple copies of a single gene. So the polymerase chain reaction is essential for community DNA fingerprinting methods because it amplifies the target genes, yes, and allow the analysis of otherwise undetectable population. So whichever population we cannot detect using this culture dependent method, we can multiply the DNA and then go for culture independent uh, identification techniques. So here both the assertion and the reason are true, and the reason like rightly explains the assertion. And the next question is, the assertion states that the next generation sequencing enables simultaneous high throughput analysis of thousands of microbial species. The reason is traditional clone library methods are low throughput and labor intensive. So initially, all this gene sequencing was done using traditional methods that include Sanger sequencing. So there it takes a lot. It takes lots of time to sequence because one base pair after the other, the sequencing happens. So it takes very high time. But in case of NGS or the next generation sequencing, it is more advanced, and it is also called high throughput sequencing because it can handle multiple samples at the same time, and it can sequence millions of DNA molecules simultaneously. So because of this parallel processing capacity, we call the next generation sequencing as a high throughput sequencing technique also. uh, so the first statement is right. Next generation sequencing enables simultaneous high throughput analysis of thousands of microbial species, and that is true. Uh, Reason is traditional clone library methods are low throughput and labor intensive. That is also true. Both the statements are true. Traditional method includes Sanger sequencing, in which base pair by base pair the sequencing happens. So the traditional clone library methods are low throughput. It cannot process multiple samples simultaneously, and it is again labor intensive. So here both the assertion and the reason are true. So next question is to identify the true statements out of the following. The first statement states that RNA analysis selectively indicates metabolically active microorganisms. The clone library method provides high resolution separation of phylotypes. Third statement is laboratory conditions can easily replicate all environmental interactions, and cultivation dependent analysis enhances the understanding of microbial interactions in situ. So whenever we talk about RNA, if the gene is functional or if it is going to be expressed, then only there is this talk about RNA. And we also know that the life short life of the RNA is so short, and the detection has to be very rapid if we go for RNA analysis. So which means if the DNA is going to be expressed, then only from DNA we get the transcript of the RNA. So analysis, RNA analysis selectively indicates metabolically active microorganisms. That is true. And then the clone library methods provides high resolution separation of phylotypes. So the clone library method is the traditional method, and it indeed produce high resolution separation of phylotypes because it is very accurate. Though it is labor intensive and time consuming, it provides high resolution separation of phylotypes. So we can find minute differences in the genetic makeup of organisms, and hence it provides a high resolution separation of phylotypes. And the third statement is laboratory conditions can easily replicate all environmental interaction. This statement is absolutely false, because laboratory conditions cannot replicate the environmental interaction. That is why we have molecular methods, or this methods in environmental biotechnology, into different categories. So one of it is doing laboratory experiments, and the other is in situ monitoring. So in situ monitoring is monitoring the microbial dynamics or activity in its original habitat, whereas we have another category called laboratory conditions, where we try to replicate, but in uh, real it is very difficult to exactly replicate the natural environmental condition. And the last statement is cultivation dependent analysis enhances understanding of microbial interactions in situ. So here cultivation dependent in the sense we are going to culture the organism in a laboratory. In that case, it is not going to help us understand the interactions in situ. So if the statement is going to be cultivation independent analysis enhances understanding of microbial interaction in situ, it is true, because we'll just directly collect the microbial sample from that particular location in which we want to do the analysis, and directly we will do use techniques like metagenomics to study the microbial community diversity. So in that case, cultivation independent analysis only enhances the understanding of microbial interactions in situ, whereas cultivation dependent analysis allows us to study the growth of the individual microorganism in a laboratory setup. So that is why both C and D are related to replicating the microbial growth in a laboratory setup. So, and both the statements are false. Only the first two statements are right. So next question is a fill in the blank question. So the first statement is DNA fingerprinting by dash separates DNA fragments by their sequence specific melting behavior. And the second question is, okay, we'll just discuss the answer directly. So the DNA fingerprinting by DGG, uh, that we have discussed in the very first question of today. Um, DNA gradient gel electrophoresis separates DNA fragments by their sequence specific melting behavior. So as we saw, we are trying to melt the DNA using different different concentrations of denaturing agent in the electrophoresis gel. But we don't want them to completely denature. That is why, in order to preserve it uh, in a little bit joint state, we are using a GC clamp at one end of the DNA. So when we talk about a fingerprinting technique which works on specific melting behavior of DNA, it is going to be DGG. So the second one is dash allows the amplification of specific genes from mixed community DNA. It is PCR. So the polymerase chain reaction, it is an amplification technique. So as we all might be aware, hi Ashwini. Okay. So I had some questions before also, since I could not find anyone. It's okay, completely fine. Since I could not find anyone joining earlier, I discussed the question and the answer very rapidly. So I'll just stop for you for this from this question. Would you be ready to answer from this? Okay. So you can just start from this question only. So I was discussing about DNA fingerprinting. DNA fingerprinting is essentially a technique in which we isolate the organism, extract the DNA, and generally go for amplification, and then we go for fragment separation, and based on the pattern of fragment separation we create a fingerprint for the organism. Okay, yeah, both the answers are right. So the answer for D is also another technique. So one of the techniques you have, right. Okay, okay. So that in here are the answers. So DGG stands for denaturing gradient gel electrophoresis. So ideally what we do, we have electrophoresis gel which contains uh, different concentrations of denaturing agent. So as the DNA moves through, the denaturing extent is also varying. In order to prevent the complete denaturation, we use a GC clamp in DGG method. So a GC bond is a triple bond. So breaking that is a bit difficult. So we use a complete 35 to 40 base pairs of GC repeats at one end of the DNA to prevent complete denaturation of the DNA. And based on the fingerprint that we are generating, we can compare it to the organisms and go for a microbial community analysis. So the answer for the first question is DGG. And when we talk about amplification, universally all of us use the polymerase chain reaction for amplification of genes. So we have a primer. The main part is to design the primer based on our needs. So the primer first goes and attaches, and after that, we are also supplying the reaction with nucleotides which go and attach one by one. And for this attachment to happen, we need a DNA polymerase. Usually in polymerase chain reaction, we will be using high temperatures. So we are using Taq polymerases. So it is from a thermophilic bacteria and it will not denature at high temperature. So that is why will be using a Taq polymerase. So the primer, nucleotides and the Taq polymerase are very important in a PCR reaction, and with this we can amplify the specific genes. And the third one is the earlier traditional method of clone library creation. So we are going to clone the PCR products individually into the DNA, and then we are going to perform sequencing of that DNA. And the last one is ARDRA technique. So it stands for Amplified Ribosomal DNA Restriction Analysis. So for the 16S gene, we will be the DNA of the 16S gene. We will be preparing the primer, and then we will do the analysis, and henceforth it is called as Amplified Ribosomal DNA Restriction Analysis. So here also we will use restriction enzymes and record the fingerprint that is generated using the restriction enzymes. So the next question is a continuation of the previous one. Which of these method is not a DNA fingerprinting technique? So DNA fingerprinting ideally involves DNA extraction, amplification, fragmentation, electrophoresis, and then recording the fingerprint based on the movement in the electrophoretic gel. Which of the following techniques does not use all the steps that I mentioned? Okay. So in this about A and B we have already discussed, and we discussed it under the topic of DNA fingerprinting. So obviously they are DNA fingerprinting techniques. The third one is fluorescence in situ hybridization, and the fourth one is the automated ribosomal intergenic spacer analysis. So out of this, C is the right answer, because in fluorescence in situ hybridization, we essentially use some fluorescent probes or tags that can attach to the DNA inside a cell, and it is a imaging technique. We use microscopy, and it is an imaging technique, and it is not a DNA fingerprinting technique. We are not doing any kind of sequencing in fluorescence in situ hybridization. That is right. So the next question is, the PCR product for taxonomic analysis is typically generated using primers targeting which of the following? So when we are going to design a primer uh, for a PCR reaction, when we are doing a taxonomic analysis, which of these conditions would be generally considered? Exactly. So the PCR product for taxonomic analysis is typically generated using primers targeting ribosomal RNA gene regions, because we know that in case of prokaryotes, the 16 is rRNA gene, ribosomal RNA gene, uh, has both conserved and variable regions. So that is the key aspect that is helping us in the entire process of the taxonomic analysis. So we will be targeting the primers to the conserved region, and then we'll go for sequencing the variable region. Whereas we don't see for any environmental DNA repeat elements or plasmids, or uh, for any virulence genes. Okay. What does phylotype refer to in microbial ecology? Okay, genetic basic marker genes, right? Uh, More or less similar. So, a group of microorganisms that share the same or closely related orthologous gene, we generally refer to as phylotypes. So, orthologous genes, like what we mean about orthologous gene is, it is said that they might have descended from a common ancestral gene. So whenever there is speciation happening, if the gene is going to be uh, evolved from a common ancestor, that genes we call as orthologous genes. So in that case, the similarity between the gene sequence is going to be very close. So the organisms which share this feature are called phylotypes. A group of organisms sharing the same or highly similar DNA sequence for a specific gene. So if it is going to be completely same or completely similar, we are going to call them as the same strains. But a particular gene or a number of particular genes are very closely related or same, those organisms together we call as phylotypes. And in case of microbial ecology, we use this to estimate the diversity and relative abundance of the taxa in a community. So because these are closely related, the metabolic potential could be uh, similar, and hence it is of high ecological significance, as you have mentioned. So the next question is, describe the primary difference between the DNA and RNA based community analysis techniques. Ashwini, only DNA based technique and RNA based technique only that is there. Are you typing something else also? Okay. DNA based techniques reveal the potential or presence of microorganism in a community. Then what about RNA based techniques? What do they tell us? Okay. So you have combined both the answers into single answer. That was the mistake here. DNA-based techniques reveal the presence of microorganism, whereas RNA-based techniques tell us about the metabolic potential of that microorganism. So the primary difference that this creates is whenever we go for a DNA-based analysis, and we can reveal the presence of all the organisms present in that community, regardless of their activity, which means regardless whether they are very much active or whether they have very less participation, but still those organisms are still present. Regardless of all these factors, DNA based analysis reveals the presence of all the organisms. Okay. Whereas when we talk about this RNA based analysis, so whenever a gene is going to be expressed, only then we get a RNA. So if RNA is produced, it means further the protein is going to be produced, and then it's going to be functional. So RNA based analysis tells us about how the metabolism of the community works, and whichever organism has an upper hand or major contribution towards the metabolism, only those organisms we will be able to identify. And one more thing is RNA is also highly unstable, whereas DNA is very stable. So even then, because of that instability, also we could miss out identifying few microorganisms. But the primary difference is DNA can tell all the microorganisms that are present, but RNA talks about the genes that are expressed, and not about comprehensively all the microorganisms that are present in that. So whichever members are highly metabolically active, those can be easily identified by RNA based method. If you want to study about what is the metabolic potential, then you go for RNA based analysis. I hope that was clear. If not, please post in the chat box for me to repeat so that I can repeat. And the next question is, what characteristics make a gene an ideal taxonomic marker for community studies? So just think about what are all the genes that we call as taxonomic markers, and then think on why we are calling them. Then you will get the answer for what characteristics can make a gene an ideal taxonomic marker. Exactly. So it should be universally present. It should be of a suitable length. for us to amplify using PCR or some technique, and it should be appropriate and variable. One more key point is that it should contain conserved and variable region. So you talked about variability. The only thing that you left is the uh, conserved region. So only when there is conserved region that is present, then only we can have primers designed for it, and we can also have, we should have some commonality. There should also be some variability of that gene in all the organisms. So ideally we use 16S and 18S rRNA genes. So it is universally present across all the taxa. It should be sufficiently conserved for a broad amplification. Just to design the primer, it should be of suitable length, and then it should also contain the variable regions so that we can discriminate using the variable regions. Perfectly right, your answer was. So the next question is a match the following question. So this site talks about some methodologies like clone library method, high throughput community profiling, and physical segregation of phylotypes. So here, what techniques we use in these methods are listed. Where do we use next generation sequencing, and where do we use gel electrophoresis, and cloning and sequencing of PCR products, where do we use? Okay, so I will take it as 1C, 2A, and 3B. Exactly. So in the clone library method, we will use cloning and sequencing of the PCR products. So we have a gene fragment. We will take a plasmid vector, clone the gene into the vector, go for screening, and then sequence. And finally, we form the library. In high throughput community profiling, when we talk about something high throughput, obviously it is going to match with the next generation sequencing. And the physical segregation of phylotypes we do using gel electrophoresis. So we do some restriction digestion and then go for gel electrophoresis, and then we can do a segregation of this phylotypes. Perfect. The next one is also a match the following question. This also deals with DNA fingerprinting, tools in DNA fingerprinting and their corresponding explanations. Okay. So universal primers are the primers that amplify 16S rRNA genes from all bacterial species. Exactly. So we have the conserved region and we have universal primers which we can use in all the um, bacterial species for amplification. And in case of GC clamp is ideally 30 to 40 base pairs long, and as the name suggests, it is a guanine cytosine rich sequence because it has a triple bond. There is a triple bond between G and C. It prevents the complete DNA denaturation. Whereas the part before the clamp has to be denatured for us to identify it using the TGG. And the phylotype identification, generally we do the grouping of clones with similar restriction digestion pattern. So we identify a gene and subject it to restriction enzymes which can cut the DNA into fragments of different sizes at different location, and based on the digestion pattern, the DNA fragment pattern that we are getting, we will do the phylotype identification. And for blue white colony selection, it is a method for identifying the positive clones containing inserts. So uh, we have a plasmid vector and the gene PCR product we will just insert into the plasmid and then put it into a microbial cell and then take it for screening. So based on whether the insert has been attached to the plasmid or not, we get blue and white colonies, and then we know which cells are recombinant and which cells are not. And the fifth one is the restriction endonuclease. It is an enzyme used to digest cloned 16S rRNA gene fragments. So after this restriction enzyme thing only, we'll go for electrophoresis and have different techniques to identify and group the microorganisms based on their DNA fingerprint. Right. So the answer is perfectly right. Next is again a match the following question, and this time based on DNA fingerprint techniques only. Okay. So ARDRA methodology we do a restriction analysis of amplified ribosomal DNA. In DGG also we do restriction analysis, but it is not of ribosomal DNA. It is based on DNA denaturation profile. And clone library construction, collection of 16S rRNA genes that are PCR amplified and cloned into plasmid vectors. And melting behavior, temperature dependent DNA denaturation influenced by the GC content. And community fingerprinting means rapid assessment of microbial community composition and dynamics. True. All are correct. So the next we come to again an assertion and reason question. So in ARDRA, clones with similar restriction digestion pattern are grouped together as same phylotype. The reason is similar restriction patterns indicate highly identical or highly similar 16S rRNA gene sequences from closely related organisms. Exactly. So in amplified ribosomal DNA restriction analysis, we have clones with similar restriction digestion pattern are grouped together as the same phylotype. Um, and they also indicate that they have identical or highly similar 16S rRNA gene. Because the fundamental principle of this technique itself is that similar DNA sequences will produce similar restriction fragment patterns. We are going to use the same restriction enzymes for the sample. So if the restriction pattern is going to be similar, then the initial template is also similar, and hence we can group them together as the same phylotype and call them closely related organisms. So it is all based on the action of the restriction enzyme. Next question is, a single band in DGGE analysis represents one bacterial species. And the reason is each band corresponds to a unique 16S rRNA gene sequence with specific GC content. Okay. Uh, so here, A is a little bit confusing statement and it is false. It is not true. Uh, so a single band in a DGGE analysis represents one bacterial species is false. If the bacterial species is having a similar denaturing gradient fingerprint, or it is going to have the same fingerprint, then it is going to come at the same places, and that may not be a single bacterial species. So multiple species, generally the species which have a similar GC content can co-migrate. So during the electrophoresis process, the migration happens. But when two different DNA representing two different bacterial species, if they have similar GC content, they can co-migrate and form a single band. And apparently, we just look at the single band and say everything represents one bacterial species. But actually, it may not be the case. The reason is each band corresponds to unique 16S rRNA gene sequence with specific GC content. So if we only see the reasoning statement, there are different bands. So each band corresponds to a unique 16S rRNA gene sequence with specific GC content. That is true. When we have multiple bands, each band comes from a 16S rRNA gene sequence with a specific GC content. So the reason is true, but the assertion is a single band always represents one bacterial species. No, that is not the case. The reason why it is showing a single band may be because they have similar denaturing properties because of the similar GC content that these genes possess. So this is one of the limitations or drawbacks of using a DGG analysis. Though we might think it belongs to only one bacterial species and count it as one, it may not really be the case. So and that is why all these methods and techniques keep evolving to increase their sensitivity and analytical efficiency. I hope that was clear. You can now try answering this question. Clone library construction requires positive clone selection through blue-white screening. Blue-white selection distinguishes between clones containing inserts and those without inserts. So this question we already discussed in one of the match the following questions. So I think it is a bit easier for you to answer. Right. So here both the statements are right. So first we do the PCR amplification, then clone the insert and then go for screening. So the screening is based on blue-white selection. So whichever has been selected or has been noted with the inserts, we take them and go for the clone library construction. So the next question is, out of these four statements, which statement or one or more statements can be false, you can identify the statement. Exactly. So in ARDRA, we should use multiple restriction enzyme, because when we use one, there are high chances of getting false positives or false negatives. So in that case, we should use multiple restriction enzymes so that we get accurate results. And the third statement, both ARDRA and DGGE are community fingerprinting methods, and they enable phylogenetic profiling of environmental microbial communities. Yeah, that is why we use these techniques. And the last statement is GC clamps prevent complete DNA denaturation, maintaining partial double standard structure for band detection in DGG. So this was the purpose of GC clamps in DGG. We were discussing it from the first. But the second statement is the 16S rRNA gene contains highly conserved regions that are identical across all prokaryotic species. They also contain variable regions. So it is not only about the highly conserved regions that are identical across all prokaryotic species. Perfectly true. So next one will be a fill in the blank question. So we have almost seen the answer for this question and a few other questions also, like 2 to 3 times we have repeated the answers. I hope this is a bit easier for you. you've identified the method rightly, but the wordings are we use plasmid vector. So, okay, that is fine. And blue white colony screening or colony PCR screening. So, yeah, you identified it perfectly. The next question is DGGA separates DNA fragments on based on their dash behavior. The exact word we have seen in one of the previous question, which depends on this also is repeating. And the third blank is about the number of base pairs, which we saw in one of the match the following question. This is just to uh, test your memory without any options. If you are able to answer this, then it would be great. Okay, a little alteration in this. First one is melting behavior and depends on the GC content directly, containing 35 to 40 base pairs. So BP stands for the base pairs, so 35 to 40 base pairs of GC rich sequences. So next is again a choose a right option. Which statement best describes the relationship between environmental DNA extraction and clone library representation? Exactly. So insufficient cell lysis can lead to biased community representation, because when we go for DNA extraction, only when we are able to lyse the microbial cell, we will be able to release the DNA and then go for amplification and fingerprinting. So if the cell lysis itself is inefficient, then the community representation is going to be biased. Only the organism whose cell wall lysis was easier, only those organisms we will be able to detect, whereas we will leave the other organisms. So next question is, what as the main advantage of using RNA instead of DNA for DGG analysis. This also we have discussed. So RNA based analysis, yeah, I just interchanged the options here, but the answer you have mentioned is right. RNA based analysis reflects active microbial community, and that is why using RNA is a main advantage. Whereas RNA is less stable than DNA, and there is no difference in the banding patterns also. And RNA amplification is certainly more, not about efficiency, but it is more tedious than DNA amplification, because we have to go for complementary DNA amplification for RNA. So the next question is about the typical size range of 16S rRNA gene fragments used in DGG analysis. So what can be the ideal range of the DNA fragment size? It should neither be too less nor be too high. Exactly, 200 to 500 base pairs is a typical range. So if you could compare the advantages and limitations of this ARDRA and DGG techniques for community fingerprinting, what could be the advantages and limitations from your perspective? Okay. So in case of ARDRA, the advantages is that um, we can get a higher phylotype resolution in that, because we are going for ribosomal DNA analysis. Phylotype resolution in the sense, whichever are genetically more or less similar, we are going to identify it as into the same phylotype. Here in terms of resolution, we literally mean something like, when we are going based on DGG, if we are going to tell that maybe some five different organisms belong to the same phylotype. Since the resolution is more in ARDRA, we can identify no, not all five, but uh, some three out of that is going to be grouped as same phylotype. So that way, the resolution is going to be high in case of ARDRA, because we are using ribosomal DNA. So the basic difference between using this ribosomal and normal DNA is that during restriction analysis, the ribosomal DNA can have the capability to show very different fragment pattern, restriction fragment pattern. But the disadvantage of ARDRA is that it is very much labor intensive, time consuming, and it is labor intensive means we have lot of steps in which people have to do it experimentally. So that one point uh, gives rise to the time consuming factor, as well as it has more steps to be performed. Whereas in the case of DGG, it is rapid, and we can also analyze multiple samples together. It is kind of relatively inexpensive compared to ARDRA. It is inexpensive, and when we are going to monitor the community dynamics, this is a good technique. But the limitations include lower resolution, because in DGG we are just talking about the melting behavior, right? So as we saw in one of the assertion and reasoning question, if the GC content is going to be more or less similar, and the denaturation pattern is going to be more or less similar, then there are chances that we identify uh, some organisms to be same, but really they are different. So this is one disadvantage, and it cannot directly provide us with sequence information. And one more thing is, it can be biased towards dominant community members, whereas the less dominant community members could be ignored using DGG. So when to go for ARDRA and when to go for DGG? When we need a detailed community characterization, we go for ARDRA because it gives a high resolution analysis. But in the case of DGGE, when we need to do some comparative studies, like we are doing some treatment before that, how the community analysis community is there, and after the treatment, how the community is shifting. So when we need some comparative analysis, temporal or spatial comparison like that, then we can go for TGG analysis. So the basic difference is what kind of data both these give, and how reliable the data can be, and then what circumstances we can use ARDRA, and what circumstances we can use DGG. So this could potentially tell you about the advantages and limitations. Okay, so I think, okay, it is interchanged. We'll just directly see this question. Okay. So the PCR is essential for community DNA fingerprinting methods, and PCR amplifies target genes, allowing analysis of. Okay, I think we saw this question previously. It has somehow repeated. So the real question is this: PCR is essential for community DNA fingerprinting. Sequencing individual clones is time consuming and costly. I think this question is also kind of repeating. Okay, there is some confusion with those questions. I'll clarify it before I upload the PPT. Okay, the next question is this. Universal 16 is rRNA primers amplify genes of identical base composition. Primers bind conserved regions, yielding amplicons of equal length but variable sequence. Exactly. The base composition need not be similar. So that is what the reason is stating. The length may be equal, but the sequence is going to be variable. So out of these four statements, which of the following statements are true? Metagenetic approaches can quantify relative abundance of taxa. That is true. DNA analysis using 16S primers only target bacterial diversity. 18S primers is for eukaryotes. So that is why the second statement is false. Sanger sequencing process billions of reads per reaction. No, that is false. Next generation sequencing is that way. NGS eliminates the need for individual clone plasmid extraction. It does eliminate the need for individual clone plasmid extraction. So this thing we do only in the traditional sequencing. I hope that was clear. And the next question is fill in the blanks again, like the one we started after you joined today. We'll just see a couple of more questions and then close today's session. all of it is true. So in NGS, the process of distinguishing sequence reads from different samples by attaching unique tags is known as multiplexing. Sequencing platforms that generate billions of reads per run are examples of high throughput or next generation sequencing. Both of the terms are right. They are just synonyms. And the clone library methods rely on Sanger sequencing technique. Perfectly right. And for the last question, it is amplicon sequencing of the 16S RNA gene is an example of metagenetic approach. Right. They yield amplicons of uniform length and variable sequence. So this is the last part of today's session. Like we'll just 2 to 3 questions from this and then close. Okay. So whenever we have some gene sequence in hand, we are just going to go to a database that we already have and then try to match it to the existing database and check whether our organisms belongs to any other already existing organism, and try to match and find out the organism. So in that case, the choice of the reference database affects the taxonomic assignment. That is true. And some example of the database include Silva and green genes. So Silva is more exhaustive, leading to fewer unclassified OTUs. That is also true. So Silva is one of the databases. First of all, the reason is right. It is a more exhaustive database. So more exhaustive in the sense it has a broader coverage. Like it is a database that has sequences of bacteria, archaea, uh, as well as some eukaryotes also. Therefore, since it has the sequences from this much diversity, it has a broader scope compared to other databases. And another advantage is it is more exhaustive. That is true because uh, when it has a broader coverage, it means that very high number of organisms are being sequenced and the data is present in that database. So it has more data points with a broader coverage of organisms. And because of this reason, the number of unclassified sequences is very less in the case of Silva. The choice of reference database affects taxonomic assessment. That is also true because so now we know Silva has more data points. It has a broader coverage of organisms, and it only has fewer unclassified OTUs. So how are we able to tell this? When we are comparing two databases, we find that Silva is more exhaustive with broader coverage and more data, right? So in that case, one has lesser data and other has more data. When you are going to feed your sequence to either of these databases, in case of Silva you can get more accurate results than when you feed into other databases. So that is why the first statement is also true. The choice of the reference database also affects the taxonomic assignments. So, for example, the difference in this content and assignments mean that same sequences can be classified into different genera or families. So, this is what the exact explanation that I had written in my notebook. So, the difference is mainly based on where our gene is going to be identified. I hope that is clear. So, next question is about the rank abundance plot. Are you aware about the rank abundance plot? If not, please let me know so that I will explain this question also. Exactly. So in a rank abundance plot, ideally what we have, the x-axis is going to represent the rank of each taxa. Like from one ranking means the taxa is most abundant, and as you go further right in the x-axis, it is going to be less abundant. And in the y-axis, you will have the relative abundance of each of the taxon. So as you move along, when the length is going to increase, it means that the number of taxa are more in the community. And as you move from up to down, so depending on the slope, it illustrates the abundance. So you get both richness as well as evenness of taxa in a rank abundance plot. And it also gives you the relative abundance, which is the y-axis. We just have two more questions, so we'll complete that and close. Uh, this one and the next one. So the clustering threshold lowering from 97 to 95 increases OTU count. Reason is lower identity threshold groups more sequences together, reducing the OTUs. So here A and B are kind of contradicting. So either one should be right. So actually, the assertion is right. Clustering threshold lowering from 97% to 95% increases the OTU count. That is true. And in the reasoning part, the lower identity threshold groups more sequences together. That is also true. The only false here is it does not reduce the OTU, but in return. Okay, I just got confused with the answer. It is that A is false because when you're going to lower the threshold, many of the organisms are going to be clustered under the same OTU. So it is going to reduce the OTU count. So A is false and R is true. I just got confused with another statement. I'm repeating this. Clustering the threshold, clustering threshold lowering from 97 to 95% increases the OTU. No, when you are going to lower the clustering threshold from 97 to 95, into same OTU itself, you are going to like incorporate many organisms. So in that case, you will not have many different OTUs, but in the same OTU itself, you will accommodate all the organisms. So that is why the number of OTUs will decrease, and that is why your assertion is false. In case of reason, the lower identity threshold groups more sequences together, reducing the OTU. So this is the right statement. The reason is right. In this assertion statement, lowering the threshold from 97 to 95 will decrease the OTU count. So that is the correct answer for this question. So the assertion is false and the reason is true. The last question is in, I hope the previous answer was clear. If not, please let me know. And the last question for today is, in the applicon sequencing, an OTU represents which of the following? In case you are confused with the previous question, please let me know. I will repeat it. You're right. In amplicon sequencing, an OTU represents a cluster of similar reads. So the last one is a heat map of taxa versus samples best illustrates the taxa abundance pattern. So to represent abundance, we have a heat map. And after this, I have some content regarding the culture independent methods, the fingerprinting uh, steps in the fingerprinting technique, and also some representation about what is ARDRA, TRFLP, and ARISA. Also, as we discussed, the difference between these two methods as a table I have given. And this is the blue white colony screening mechanism or principle, and also a simple difference between Sanger sequencing and new generation sequencing. So this is just to highlight the topics that has been covered. So you can go through this once I upload the PPT. And thank you for joining for today's session, and thank you for answering for all the questions. If you have any doubt, please let me know. Okay. Thank you so much, Ashwini. Can I close the session? Thank you so much. See you next.